Hello, Biology 1 students. Mitch Albers here from the Biology Department at Minneapolis College. In this lecture presentation, we're going to cover Chapter 14, which is titled Mendel and the Gene Idea. Chapter 14 is on Gregor Mendel, and Gregor Mendel has been kind of given the title as the father of modern genetics. Mendel was studying at the University of Vienna, and he had two mentors who really kind of played a crucial role in molding who he became and how he became the father of inheritance, if you will. One was a Professor Doppler, who was a physicist, and he really got Mendel trained and focused on using mathematics in his work. And another plant biologist named Unger had kind of steered Mendel into working with plants. And so Mendel combined the idea of using mathematics to look at inheritance patterns in plants from generation to generation. And this is going to be one of the things was significant in how he presented his data and also how it was received back in his time. If you go back to the mid-1800s and if you kind of asked around, what did people generally think in terms of inheritance? How were traits inherited in the offspring from their parents? Well, the basic idea back then was the blending hypothesis idea, and that is as simple as finger painting in kindergarten, where you take two paint colors and mix them together and you get a blend, right? And so the idea is that offspring are simply just a blend of traits from their two parents. And that makes sense. And however, Mendel challenged that idea. And so he kind of went against the paradigm, and his experimental results showed that traits aren't necessarily a blend. And we'll get into the details of how he figured that out. So again, if we want to think of blending inheritance as the major mode of inheritance, right? So use that fingerprinting analogy. If you combine red with white, you're going to get a pink color. If you combine black and white, you're going to get an intermediate. You're going to get a gray shade of color. And so that's that blending idea. And when you look at how offspring's traits show up, that not every offspring is a blend of their two parents. Okay, and Mendel saw this out in nature. He saw that parents aren't a blend, that they show different traits. So again, if you take white and combine it with red, you're going to get an intermediate like pink in the middle, and getting that blend between the two parents in the offspring. Mendel didn't see it that way, and his results again will show that for many traits they don't follow that blending idea. And certainly if you look at offspring, a lot of times the offspring look like maybe just one of the parents and not both parents equally, right? So this kind of intrigued Mendel, and so he set up his experiments to demonstrate that they necessarily don't always occur by blending their traits from the parents. Just examples, okay, where you've got offspring here, and oftentimes the offspring more or less resemble one of the parents and not both parents equally. And certainly, every time we bring home, new parents bring home a new child, that's what everybody does. They're looking at to see who nose did they inherit, whose eyes did they inherit, whose hair did they inherit, right? We're looking for those traits from the two parents. But again, sometimes some of the traits look like maybe just more of one of the parent and not both parents. So why is that? Well, let's try to figure that out. So let's kind of, again, back in the mid-1800s, okay, what was known? We just got done talking about cell division, mitosis and meiosis. And when Mendel was doing his work, he had no idea about the concepts of mitosis and meiosis. It hadn't been figured out for another 40 years after Mendel. The late 1890s is when mitosis and meiosis were really worked out. And the idea of chromosomes was just coming into an understanding in the early 1900s. And certainly we didn't understand, we didn't even know the structure of DNA until the 1950s, when Watson and Crick figured out the structure of DNA in 1953. And when they figured out that structure, that was when all of a sudden it became apparent how the structure and function of a gene was. So, 100 years before we actually figured out DNA and genes, Mendel had figured out these patterns of inheritance long before these things were really absolutely determined and figured out. One of the things you're gonna discover in studying Mendel is that when he had published his results, again using mathematics, a lot of people really didn't understand his work, and nobody really appreciated the work that he did during his time. And so sometimes we say that Mendel maybe was way ahead of his time. He was more advanced in figuring this stuff out before some of these other things have been known. And the cool thing is that once, like mitosis and meiosis had been figured out, and once we had figured out chromosomes, DNA, and genes, that that new information that came about later on after Gregor Mendel's work only validated that he had got it right back then. Mendel, again, working around in the mid 1800s, he was working in in a monastery. He was a monk, so he's associated with the church. Like many people, many scientists, prominent scientists back at the time, were also associated with the church. And he was working with garden pea plants in a courtyard garden space next to the abbey that he was a monk at. And anyhow, he chose garden pea plants for a number of reasons. Very identifiable characteristics, and I'll get into those in just a moment. And again, he set up extremely controlled experiments, made accurate observations and detailed notes about his experimental work, and he was methodical. And then the data that he kept, he used numbers, he used fractions, he used percentages, he used ratios to explain the results that he had seen with these garden plants in terms of how the traits were being passed on from parents to offspring. Okay, so the first thing to note here is that here's a notable trait, is the color of the flowers. In the pea plants, he saw that it varied. They're either purple colored flowers, or they were white colored flowers. It was one or the other. And what he could do in experiments is he could control the crossing, the breeding of these plants, and that's why he could follow these traits and see what were the traits of the parents, and then look at the traits of the offspring. So he used a paintbrush to transfer pollen from the male parts of the flowers to the female parts of the flowers through the process of pollination, and then he could observe what was the results in the next generation. So when we are studying genetics, the F stands for the word filial, which means offspring. And F1, this is the first generation of offspring from a set of parents. And so these two plants that are being crossed here, the purple flower with the white flower, are part of what's called the P generation. The P stands for parental generation. Okay, so for example, you are the F1 offspring of your parents. If you have kids, your kids are the F2 offspring of your parents. They're the second generation as the grandkids. One of the reasons Mendel found garden pea plants easy to work with is that they grow very quickly. Their life cycle is fairly short. He could control them in their crosses. And they had some extremely observable traits. And so he followed 7 different characters in these plants, from the flower color, flower position, seed shape, pod color, length of the stem. And those are all shown over here in this table. So let's take a look at those in a little bit more, a little more close up. So just like I showed in the previous slide, he looked at flower color, and he saw, a variation from purple and white. And then the other thing that you're going to see, and as an example of him using mathematics, right, is that he did many crosses of these traits and these plants to generate data. So these are the number of offspring that he had observed when he crossed purple with white. And then he took it from the P generation, parental generation, to the F1 generation, and then he crossed the F1 generation with each other and took it all the way to the second generation, the F2 generation, and that's the numbers we're looking at here. What did these numbers look like in the F2? Well, and then when he took these numbers, he simplified them into a simple ratio. Now I want you, as we kind of go through all these, I want you to see a theme here with the ratio, is that if this is pretty much just a 3-to-1 ratio. And when we look at these other traits, you're going to see that that is consistently the same with all the different characters that he traced for inheritance, that they showed in the F2 a 3-to-1 ratio. Here, looking at the position of the flower, axial flowers were ones that branched off the main branch before the end of the main branch. Terminal flowers are the ones found at the end of the main branch. Again, he hundreds of plants, and he looked at the F2 ratio, 3-to-1 ratio. Seed color. He saw that the varied traits were either yellow seeds or green seeds. Here he looked at thousands of outcomes, and again, when he simplified the ratio, 3-to-1 ratio. Same with each shape, round versus wrinkled, thousands of offspring. Again, 2.96 to 1 ratio, that's a 3-to-1 ratio. Same with seed pod shape, pod color, they all came out as a 3-to-1 ratio. And then the last trait that he looked at was the height of the plant. And tall, outnumbered the short or dwarf plants in a 3-to-1 ratio. 2.84, you round that up to a 3-to-1 ratio. We'll come back to this 3-to-1 ratio in just a moment. So let's just take a general look at what Mendel had saw in his experimental results. Now we'll just focus on flower color, okay? So when he crossed as the P generation, when he crossed a purple flowered parent with a white flowered parent, okay? So this is our P generation, they're our parents. He started with what are called true breeding parents. And what he meant by true breeding is that for the purple flowered parent, when that was crossed with other purple flowers, they only produce purple flowered offspring. So he chose one of those plants to be the parents. Same with the white, that it was true breeding white parent, that if he crossed other white flowered plants with this, they only produced white offspring. And so he picked one of them to be the starter parent here, as being a true breeding white flowered parent. So when he crossed the purple with the white flower in the P generation, the F1, the first generation of offspring, were all purple flowered. Okay, so the white trait completely disappeared in that first generation of offspring. And this is where he came up with his law of dominance. That purple was dominant to white. And white now, having disappeared, he referred to as the recessive trait. So with those F1 offspring, okay, he allowed them to self-pollinate or cross-pollinate. And when he crossed purple with purple in this generation, and when he did that in the second generation, then he found the white trait showed back up, but not in an equal proportion. Again, he looked at this out of, let's say, 4 offspring, and you can see here there's 3 to 1. There was a 3 to 1 ratio of purple to white. So that theme kind of played out when he looked at these various traits in pea plants, that one trait was dominant to the other trait, which was recessive for the different characters. Now, keep in mind that Mr. Mendel did not know how cells divided. The technology wasn't available yet to see in the cell, to see the chromosomes, and no knowledge of what the genetic material was in the cell. He had no idea. However, he used a term called factors. Sometimes you'll hear it described as his little factors. That there was this unknown, unseen thing that must be in cells. Today we know it as genes, but Mendel called them factors, and these were unknown. He didn't know anything about them, but he said there's got to be something there. Again, he used the word factor. There's some factor in the cell that's controlling how these traits are being inherited from parents to offspring. Well, today we know that we get a set of homologous chromosomes, and half come from dad, half come from mom. The blue would be the paternal chromosome, the red, the maternal chromosome. And each parent passes on what we're going to refer to as an allele for a specific trait. Okay? Now that term allele, we're going to refer to as an alternate form of a gene. And you can see that being shown here is that both mom and dad don't always pass on the same genes. They both have, in this instance, the gene for flower color, but the father has the purple allele and the mother has the white allele. So we see different variations in the gene. And based on the previous discussion with purple and white flowers, we said the purple was the dominant trait and the white was the recessive trait. So again, Mendel didn't have any visual model like we have here. He didn't know what an allele was. He didn't know what a gene was. He didn't know what a chromosome was. He didn't know what DNA was. He just said that they were these little factors within the cell. An analogy of alleles, right? Variations in a gene. Well, like with ice cream, there's variations in ice cream too, right? There's not just vanilla, there's chocolate, there's strawberry, and lots of others. There's lots of variations, and we see that happening with genes as well for certain traits. Now with his garden pea plants, he saw variations that were one or the other, purple or white, tall or short, yellow seed, green seed, that sort of thing. Now, let's go back to our original example here where he crossed the purple flowered plant with a white flowered plant. Now we know that purple was dominant. So we're going to use letters to represent dominance, and capital letters always equal dominance. Small case letters are going to represent recessiveness. So for the in the P generation here, this is the genetic makeup of the purple flowered parent, big P, big P. So remember, this parent had to have received the dominant purple flower from, let's say, its mother, and it also received a dominant purple gene from its father. And that's how that genotype for that individual came to be. Same with the white. Now the white, these are small letter P's, so white is recessive. And this parent, who also had parents, okay, both the mother and the father both passed on those small p's. And that's why this genetic makeup is for that parent. And these are what we call the genotypes. Using those letters to show the specific genetic makeup for the trait. When you see the trait and you describe it, the outward appearance of that trait, purple, the outward appearance of that trait, white, those are called phenotypes that describe how you see the trait. So your hair color, your eye color, how tall you are, how you see that trait, those are your phenotypic characteristics. Just like that list of 7 things that Mendel was following in his garden pea plants. Those were the phenotypes, and these are the genotypes. So how do we display the genotypes running this cross like we did in the previous example? So the 2 parents, one being purple, the other being white, when they cross, all the F1 generation were purple, okay? And here you can see that the genetic makeup for the genotype is showing a dominant with a recessive. And what we're saying then is this dominant allele masks that recessive allele, and the dominant trait is what is exhibited. You see that dominant purple trait. This genotype can be described where both letters are different as being a heterozygote. Or heterozygous, G-O-U-S, is the condition. And basically what that means is if you look at this, here are the 2 parents, here's the mother, here's the egg, here's the father. Okay, in plants this would be pollen, but it'd be analogous of like say sperm in animals. Well, those 2 gametes come together, the egg and the sperm, and you get big P, little p, you get a heterozygote. So then what did Mendel do? Well, then he said, all right, let's take 2 offspring from this F1 generation and let's cross them. Big P, little p with a big P, little p. 2 heterozygotes we're going to cross. So let's move on to the next generation here. And that's what we see in the F2s. So now we're using a tool here called a Punnett square. Which will show us the phenotypes and genotypes. So the way that we do this is we take our 2, so since both parents, right, our F1 parents are now, our F1 genotypes were heterozygous, big P, little p, crossed with a big P, little p. And so one is the mom, one is the dad. So we're going to separate this and to say this is the mom over here, and these are the gametes, okay? So remember, in meiosis, okay, the parent here is diploid. We have to produce haploid gametes. And we do that through meiosis. Same with the father. He's going to go up here on top. So this is the father. And since he had the same genotype, half his gametes are going to be dominant, the other half are going to be recessive, big P, little p. And these are haploid. Again, the father had to go through meiosis to produce those gametes. Okay, and then what we do is we just say, all right, well, again, let's say these are sperm or pollen, which are analogous to sperm in plants. What's the probability of this egg getting fertilized by these two sperm? So you just bring the big P over and bring the big P down, and now in this, you're looking at a possibility out of 4 offspring. This offspring is going to be big P, purple. Okay, just like the original parents were in the parental generation. Bring this P over and this little p down, and then you're going to have big P, little p, a heterozygote, just like the parents. Same here. Bring the big P down, bring the little p over, and you produce another heterozygote, but it's purple. So these two heterozygotes are purple. The dominant overrides, masks the recessive trait. And then we see little p, little p. This is where that recessive trait shows back up in the second generation, and our genotype is little p, white. And that, again, that trait shows back up in the second generation. So it wasn't lost in the first generation, it was just masked or hidden, and then it shows up here. Now if we do the analysis, when Mendel did this kind of cross, he then saw phenotypically, phenotypes, the appearance, he saw 3 purple to every 1 white. And we get that typical 3-to-1 phenotypic ratio. Okay, another ratio he also kept track of was the genotypic ratio. And here we have 3 different genotypes. We have big P, big P, little p, and little p, little p. And out of the 4 offspring, there was 1 of these, there was 2 of these, and 1 of these, which gives us a ratio of 1 to 1 for our genotypic ratio. That's a good example of Mendel now demonstrating that blending inheritance isn't the mode of inheritance, that there are traits that are dominant and are recessive. And yeah, so, and now he used these numbers to kind of show the results that he got. Here's another example, just a quick one. When you cross a tall plant with a short plant, again, they're true breeding, big T cross with a little t, they're going to all produce offspring that are big T, little t. Now, they're not an intermediate height plant. They're not midway between tall and short like a blend would, but they're exhibiting the dominant trait, tall. These F1 offspring are tall and not dwarfed or short. Okay, but when Mendel crossed offspring of the F1 generation, which were heterozygous, big T, little t, with each other, the final result is that he got that 3-to-1 ratio of 3 tall, right, to 1 short. Okay, just another example. Let me clear that off. Here's another example that we can use here is albinism. Is the inability to produce the skin pigment in your skin, melanin. And albinism is what we refer to as an autosomal recessive trait. Now that term autosomal will play out more when we get into chapter 5, but it's a recessive trait. So you have to be, let's say, small a to exhibit albinism. So what happens if, let's say, you have parents who are normal skin color, they have melanin, but they carry the recessive trait. So if you cross, this is our P generation, and both parents are heterozygous, but they carry the recessive albinism trait, right? So when we run this through a Punnett square, say this is the female, this is the male. Okay, the female is going to produce, remember these are diploid conditions. They're going to produce haploid gametes. So let's say these are the eggs for the female. So half are going to be the dominant normal skin, half are going to be recessive for albinism. The father is the same thing. So this is big A, little a. So let's just, we'll use sperm as an example. Then when you bring them together, big A, big A, big A, little a, this looks pretty similar to the one we just did, just using different letters, different traits. Little a, little a. You know, so what happens if both parents are carriers for the recessive trait? What's the probability of them producing an offspring who's got the recessive condition, albinism? Okay, well, that's going to be 1 out of 4. 1 out of 4 offspring will have albinism. Or 1/4, or 25%. Again, just kind of using an example of using mathematics to represent the outcomes of these crosses, these traits being passed on from parents to offspring. So that's just, again, looking at where, you know, normal skin pigmentation is the dominant, recessive is albinism, just running that through a Punnett square as another example. Let's just play around with some of the terminology here a little bit more. Just reinforce these. Phenotype is the outward appearance, how we see the trait. Okay, that's what we're seeing, that's what we're seeing, that's what we're seeing, that's the phenotype. Just like if you look into somebody's eyes and you describe their eye color, blue eyes, brown eyes, hazel eyes, that's describing the phenotype. The genotype is the genetic makeup. Big P, big P, big P, little p, big P, little p, little p, little p. When the two alleles are the same, the word that describes that is homozygous. If the two alleles are the same and they're dominant, we'll describe this genotype as homozygous dominant. Heterozygous is heterozygous. You have one dominant and one recessive. Okay, one dominant, one recessive. Heterozygous, hetero meaning different, homo meaning the same. And then this last example for the white phenotype, then they're both little p, little p. Again, that's a homozygous, but then this is going to be homozygous recessive as the trait. And what we saw in that previous example, we had a phenotypic 3-to-1 phenotypic ratio. And then we looked at genotypic ratios, and there was one homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. So we had that 1-to-1 genotypic ratio. We could ask the question here, how do you know what the genotype is? Because For purple flowers, a purple flowered plant could be this or this, a homozygous dominant or a heterozygote. They both will display the dominant phenotype. Well, you can do something that's called a test cross. Whoops, test cross. And in a test cross, you're gonna say, well, the unknown here is the purple. And if we want to determine is it homozygous dominant or heterozygote, we're going to test cross with the unknown with a homozygous recessive parent. In this case, little p. And that will tell us whether we've got a homozygous dominant parent or a heterozygous parent. And so here's how that works. Let me clear that off. So if the parent was a homozygous dominant parent, big P, and we cross it with a parent who is homozygous recessive, well, then we're going to get all our offspring are going to be heterozygous purple offspring. So if all of the offspring end up being purple, well, then we know we're dealing with a homozygous dominant parent. Okay, that answers the question. If we do the cross and half of the offspring are dominant and half of the offspring are homozygous recessive, little p, well, then we know if we see any white in the cross, then we know that the parent was big P, little p, a heterozygote. And that's just shown here down at the bottom of the screen. Okay, so if you're not sure whether it's a homozygous dominant, heterozygous parent, you run a test cross by crossing the unknown genotype. That'll tell you which genotype you're dealing with. Now let's move on to where Mendel was working with two traits. This is going to be something called a dihybrid cross. When we dealt with one trait, it was a monohybrid cross, mono meaning one trait. Here we're going to be looking at two traits for our example. So here we're looking at the two traits are going to be the color of the seed and the shape of the seed. So seed color and seed shape. Now, just like we did with the monohybrid examples where we were looking at a single trait, the P generation is going to be true breeding for two traits now. So this parent here is going to be yellow and round. The other parent is going to be green and wrinkled. Now, yellow and round are the dominant traits, and they're represented with capital Y and capital R. The recessive traits, green can be a little y, and wrinkled, little r. And so both parents here that we're going to cross are homozygous for the two traits. Homozygous yellow, homozygous round, crossed with a parent who is homozygous recessive, green, homozygous recessive, wrinkled. When we do this first cross, remember these are diploid conditions, and we have to produce haploid gametes. We have to reduce our chromosomes number to 1/2. We're going to do that by meiosis. Okay, so for both parents to produce gametes, they go through meiosis, and so this parent here can only produce big Y gametes and big R gametes, and that's why we see this showing up in the gamete here. This parent here can only produce small y and small r gametes, and that's what's showing up here. And that's how we get our haploid gametes. So we'll move on and then look at the F1 generation here. So the F1 offspring are all heterozygous for the two traits. So they're all heterozygous, yellow and round. So they're exhibiting the dominant traits. The recessive traits are hidden and masked. They're not lost. These offspring are now carrying those recessive traits. Now, one of the things that Mendel had discovered was all the traits in these garden pea plants were inherited independent of each other. In other words, round or yellow didn't influence round, green didn't influence wrinkled, yellow didn't influence wrinkled, and green didn't influence round, meaning that the seed color didn't affect the outcome of the seed shape, that those traits are inherited independent of each other. And that's exactly what we're going to see happening here. Now, had the traits not be inherited independent of each other, we would see this outcome where if we went through dependent assortment, where the way that the alleles segregate out is just this combination, which we'd call dependent assortment, then we would get one outcome. But what Mendel had determined is that these alleles assort independent completely, okay? So for instance, this parent here is going to produce more than just two types of gametes. It's actually going to produce four. And if you recall from mathematics, algebra, you probably remember using a FOIL method for looking at genetic traits, okay? And you should remember F stands for first, O stands for outside, I stands for inside, L stands for last. And what we're going to do is we're going to FOIL this, because that's what meiosis is going to do, okay? So in other words, we're going to look at what are all the possible gamete combinations, and there's four of them. So we do the first, and we're going to do between the Y's and the R's. Big Y is first, big R is first for seed shape. Okay, then we do the outside, which is big Y, little r. These are the two on the outside. Then we're going to do the inside, little y. These are inside, little y, big R. And then we do last. Little y's last and little r's last for the two. Little y, little r. So these are the four possible gametes that are now, these are all haploid gametes coming from this diploid genotype. Okay, so let me erase that quick. Mendel had determined that independent assortment is how these things work. However, he had no idea that meiosis was the process that did this independent assorting. But he had, in his estimates and his observations, determined what we call his law of independent assortment. The idea of dependent assortment is not the correct process that's going on here. What we see then here is now using a Punnett square, looking at the probability and what the offspring look like out of 16 offspring for 2 traits. To work out this problem like you're seeing here, this is the one that's out of your textbook. Let's just work this from scratch. So we had a big Y, big R, little r, a yellow round parent crossed with the exact same genotype and phenotype, a yellow round parent, but they're both heterozygous for the 2 traits. And these are, again, these are the diploid conditions. Okay, so when we do this cross, each of these parents have to produce gametes through meiosis. And again, then let's just use that FOIL method. What are the firsts? Big Y, big R. What are the outside? Big Y, little r. What are the insides? Little y, big R. What are the last, the L's? Little y, little r. So if this was the female, these are 4 haploid eggs that get produced through independent assortment. For the male, do the same thing. Foil the male here, and we're going to produce big Y, big R for firsts. What are the outside? Big Y, little r. Outside. What are the inside? Little y, big R. And what are last? Little y, little r. These are now going to be the gametes, and I'll just draw some sperm here. These are the haploid sperm, or if they're in plants, pollen. Now we're looking at setting up a Punnett square that's going to look at what is the probability of the genotypes and phenotypes out of 16 offspring. Okay, so let's go ahead and set that up. This won't be perfect, but it'll work. So again, we'll put all the eggs over here, big Y, big R, big Y, little r, little y, big R, little y, little r. So these are all eggs. So this will be the mother. On the top here will be the male. And we're just going to line up these sperm along the top. So big Y, big R, big Y, little r, little y, big R, and little y, little r. And we're basically looking at what is the probability of these 4 sperm fertilizing these 4 eggs. And we just do the same thing. We bring the Y's over and down, big Y, big Y, big R, big R. Bring them over, big Y, big Y, big R, little r. Again, I'm just bringing them over and down. I'm keeping the Y's in front of the R's, and I'm always keeping the dominant in front of the recessive. That's just a rule. Okay, big Y, little y, big R, big R. And we're going to have big Y, big R, little r. A heterozygote just like the parents were. Okay, do it again. Now for the next row. Big Y, big R, little r. Okay, big Y, little y, or excuse me, big Y, 2 little r's. Big Y, big R, little r. Another heterozygote, just like the 2 parents. Big Y, 2 little r's. Okay? Big Y, 2 big r's. Big Y, big R, little r. Another heterozygote like the 2 parents. 2 little y's, 2 big R's. Okay, 2 little y's, big R, little r. Okay, and last row here, big Y, big R, little r, big Y, 2 small r's, 2 little y's, big R, little r, and then 2 little y's and 2 little r's. So we would line all these up. These are all our genotypes, big Y, big R, little r, or big R, okay? That's this one right here, okay? So I'm just gonna circle these when we check them off. How many are there out of the 16 offspring? There's 1 out of the 16 of those with that genotype, and they're yellow and round. Then I'm going to look at the next one here. Big Y, big R, little r. How many like that in the 2 of those, or 2 out of the 16? Let's look at this one here. Big Y, little y, 2 big R's. Okay, 2 of those, 2 out of the 16. Then we've got the heterozygotes. 1, 2, 3, 4. Big Y, big R, little r. There's 4 of those, 4 out of the 16. Then we've got big Y, 2 little r's, yellow and wrinkled. There's 1 of those, 1 out of the 16. Okay, and then we have big Y, 2 little r's. 1 of those, there's 2 of those, 2 out of the 16. Then we've got little y, 2 big R's. Green and round. 1 of those. Okay, and then we've got green and round, but heterozygous round. Big R, little r. That one and that one. 2 of those. And then the last but not least, homozygous recessive for both traits, little y, little r. Green and wrinkled, 1 out of the 16. Add this all up, we should have 1 plus 2 is 3, plus 2 is 5, that's 9, 10, 11, 12, 13, 14, 15, 16. We got everything accounted for, 16 out of 16 offspring. These are all yellow. Okay, and these are all round. So yellow, round offspring. Add these up, 1 plus 3, 4, 5, plus 4, that's 9 are yellow and round. Okay, now we're looking at what are yellow and wrinkled. Yellow and wrinkled. 1 plus 2, that's 3. 3 of those. Okay, now we're looking at how many are green, and wrinkled, or green and round first. Green and round, green and wrinkled. Okay. Green and round, 2 plus those are 3 of those, and then we just have the 1 green and wrinkled. 1 out of the 16, 1. So we end up with a phenotypic ratio and this is what Mendel found with all those traits. If he crossed 2 different traits with these garden pea plants, they always showed a 9:3:3:1 ratio if both parents are heterozygous for those two traits. You will always get this 9:3:3:1 ratio, and that's what we see here, 9:3:3:1. That will always be the case. So now imagine if you had a tri-hybrid cross, 3 traits. Well, then your Punnett square would be like 64 offspring. They get kind of crazy. So this is as far as we'll have you work out any Punnett square problems, is with a dihybrid cross. Again, Mendel didn't use a Punnett square. He did it all mathematically. And J.C. Punnett, who introduced the Punnett square, did so in the early 1900s, so long after Mendel had done all his work, just as a good visual tool to show what's going on with the cross. Anyhow, that's a dihybrid cross using Mendelian pattern of inheritance with dominant recessive type traits. So, and that was just, this is just explaining that last diagram we were looking at a moment ago. So let me, let's clear this out. And we'll move on to the next slide. There's just a little cartoon. All the fellow monks at the monastery are a little fed up eating peas. Okay, the last thing I'm going to show you is how this is going to go right back to our discussion at the beginning, is that we can't just say that all traits don't show a blending. And this next one, called incomplete dominance, if there is a pattern that supports the blending idea, this would be it. In snapdragons, we see red, we see white, and we see pink variations in the flower color, in the petals of the flowers. What we're going to show you here is we're going to use the letter C just to represent color. So the genotypes here, in order to be red, you have to be homozygous, big R, red. To be white, you're going to be homozygous, big W. So we're going to just say that these traits are in the superscript, are linked to those chromosomes. So when we cross a red flowered snapdragon with a white flowered snapdragon, now we're going to get that blending happening, just like finger painting. But remember, these are diploid genotypes, and these parents have to produce haploid gametes. Okay, where we only have half the number of genes in the gametes. So when you mix them together, then you're going to produce this genotype, which is a blend and is pink. The phenotype is pink. The genotype for pink is a red mixed with a white. And if we do the same kind of thing that Mr. Mendel did, is if we cross 2 individuals in the F1 generation, so now we're going to cross a pink parent with another pink parent, then that's what we're looking at in the F2. Unlike what we saw when we looked at a trait that follows a typical dominant recessive pattern, like with the flower color in pea plants, here we're looking at the genotypic and phenotypic ratios being different, because this trait isn't following in snapdragons a dominant or recessive pattern like we saw in the pea plants. Okay, so again, these are diploid parents, and we have to produce haploid gametes. So half the gametes are going to be this, half are going to be this. Okay, so for both parents. So we're going to put those gametes in the Punnett square. And when we run that, then we're going to bring this R over and this R down. So here the two R's come back together and we get a red snapdragon. When we bring the W and the R over, then we get an R and a W, then we're going to get a pink offspring. Okay, W over R down, we get a pink. And then W, W, and then a white showing up in the second generation. Now, what we don't see, like we saw with the Mendelian examples earlier, is we don't see that typical 3-to-1 ratio in the F2s. We have now, for the phenotypes, we have a red offspring, we have 2 pink offspring, and 1 white offspring. So there's 1 of these, 2 of these, 1 of these. So for the phenotypic ratio, it's going to be 1, 2, 1. If we look at the genotypic ratio, then we're going to have one of those, 2 of those, 2 pinks, and 1 of those. And so our genotypic ratio is going to match our phenotypic ratio, and they're going to be the same. That's when we're looking at where the trait is incompletely dominant. And again, if there's anything that exhibits a blend, that's going to be incomplete dominance. So now in humans in particular, we have most of our traits follow a Mendelian pattern, Mendelian dominance, recessiveness type pattern. However, there are other patterns, and in chapter 14, where you're introduced to Mendelian genetics, you're also introduced to some other modes of inheritance, like we did here with incomplete dominance. So chapter 14 also discusses things like codominance, multiple alleles, pleiotropy, epistasis, polygenic inheritance. Okay, you're welcome to read about those and look those things over, but I will not be testing you on those patterns of inheritance. So those of you that are going to be biology majors, you will be taking a genetics class as a core course for your biology program, and you'll get into those modes of inheritance, whereas in Biology 1 we don't cover those, but I know your textbook does. So when I come out with the study guide for exam 3, you will not be responsible for knowing those patterns of inheritance, but they're extremely important. For example, like your blood type is controlled through what's called multiple allele inheritance. And so you might want to just kind of look that stuff over, but again, you won't be graded or tested on that. All right, we are through chapter 14 and are ready to go on and talk about chromosomes specifically. So we'll see you in another lecture here on chapter 15. With that, we'll see you in the next lecture.